Hello, and thank you for joining us for the Meeting on the Med BioCentriq presentation. My name is Silva. I am the Director of Business Development at BioCentriq, a full-service cell and gene therapy CDMO located in New Jersey, in the USA. The highlight of this talk today is to introduce BioCentriq to all of you and its capabilities, and talk about a unique hybrid model that's differentiated us among peers. BioCentriq was established in 2018 to focus on small to mid-sized biotech companies to address the development and manufacturing challenges they face. One major challenge is getting access to good quality GMP facilities in a reasonable amount of time. As many of you already know, only 5 to 10% of required CDMO capacities exist today, and biotechs looking for outsourced cell and gene therapy process development or production face up to 18 to 24 months of wait time just to get into the facility, especially if you're a small to mid-sized biotech. Even when you do eventually get into a GMP facility, small clinical stage clients are typically given lower priority by CDMOs. And also, there's challenges with visibility when it comes to contracting with a CDMO, which generally leads to longer and unexpected delays. These factors create a lot of risk and leave room for costly errors. We established BioCentriq for these reasons. In the last few years, BioCentriq has grown rapidly by over 300%. We are currently working on manufacturing multiple therapies in the GMP facility. And we're also expanding, adding more than 9,000 square feet to our existing footprint. BioCentriq currently has 2 facilities in New Jersey, one in Newark, New Jersey, which has 2 ISO 7 GMP manufacturing suites, quality control, as well as analytical suites. BioCentriq's South Brunswick site has 2 ISO 7 GMP manufacturing suites, as well as our BSL-2 certified process development lab, which is used for all tech transfer activities, quality control, analytical assay development, and qualification activities. BioCentriq currently has available capacity for new projects in the process development and GMP manufacturing stages, and we typically initiate projects within 30 days of contracting. All IP generated at BioCentriq's facility is assigned to our clients. And lastly, we operate under a hybrid team model, which is unique to BioCentriq. Hybrid team model allows your development team and your SMEs to be on site at both our process development facility and GMP facilities as required. When thinking about a GMP project, that allows for uniquely joint quality oversight on the day-to-day activities. We will provide the adequate GMP training and qualification so that you can be a part of our manufacturing team. The hybrid model has been great for virtual companies who are looking for hands-on contributions to their project, as well as companies with limited GMP experience willing to learn and gain hands-on experience by using a contract CDMO. It's been also great for cell and gene therapies, which inherently have their own risk challenges built in. BioCentriq's focus is in cell and gene therapies. In the cell therapy space, we work on autologous and allogeneic cell therapies, namely T cells, NK cells, stem cells, epithelial cells, dendritic cells, and others. In the gene therapy viral vector space, we can work with both suspension and adherent systems, typically within lentivirus, adenovirus, and AAV systems. We also have an analytical development suite, which we use for assay development, assay transfer qualification activities for all of our projects. As I mentioned before, BioCentriq offers clinical manufacturing and process development as their main services. In clinical manufacturing, we offer phase 1, 2, and 3 production. We're a fully compliant GMP facility with electronic quality management systems and fully trained personnel. In process development, we perform all tech transfer activities, process design and scale-up, and analytical method development and validation activities in our process development suite. We have experience performing multi-scale, multi-platform feasibility studies. We are fully equipped as a pilot plant and have a variety of applications and technologies that clients can work with. We also do analytical development and have R&D expertise on staff. Our GMP facility is designed as a multi-product facility. Each suite has their own air handling system, which minimizes cross-contamination. The facility has an FDA-compliant MES QMS systems up and running, as well as controlled access and multi-layer firewalls to prevent unauthorized access. We also have an environmental monitoring system built in that monitors the facility 24 hours a day. Additionally, we have on-site QC and analytical support, cryostorage stability chambers, as well as backup power for the entire facility. Our current GMP footprint is about 12,000 square feet with four GMP suites supporting our operations. All our sites are ISO certified, FDA registered, and we use Master Control as our QMS, Handwell as our remote equipment monitoring system, as well as Quantus environmental monitoring systems up and running. Here are some highlights of our processing systems in our GMP facility. We use both suspension and adherent systems with scales ranging from anywhere from 2 liters all the way up to 200 liters in suspension culture. In cell therapy, we use a variety of platforms to perform open system manipulations as well as closed semi-automated unit operations. Quality is an important consideration in BioCentriq's operations. We use a 6 systems approach for quality, which includes the production systems, facilities and equipment systems, lab controls, material systems, as well as packaging and labeling systems. The facility is FDA registered and 21 CFR 211 compliant. BioCentriq has over 2,000 square feet of process development space, which includes a wet lab space for upstream development, bioreactor scale-up, as well as PCR and downstream purification activities. In the upstream wet lab space, we have equipment for upstream analytics, including the ViaSol cell counter, a flex tube tablet analyzer, and microscope systems. We also have plate readers for UV-Vis, fluorescence, and luminescence-based assays. Additionally, we also have fluorescent microscope, a DNA hood, and a ChemiDoc imager for Western blots and electrophoresis. On the top right, we have the bioreactor scale-up lab, where we currently house the Paul STR50 single-use suspension bioreactor and a set of biostat BDCU twin towers by Sartorius for running 2-liter bioreactor systems. BioCentriq also has a PCR-based analytics lab where we can run qPCR, ddPCR assays. We also have additional space where we have installed our ISLS 500 bioreactor, as well as our STR200 bioreactor. In our downstream processing lab, we have multiple platform shakers, number 15, as well as 2 bio-SMBs for continuous chromatography. We also have flow cytometry systems, a Repligen cross-flow KR2i tangential flow filtration system. We also have flow cytometry systems and multiple TFF and downstream processing units. And you can see our process development equipment highlights. Let's look at our analytical development capabilities. We have outstanding analytical capabilities that enable us to characterize cell and gene therapy products that we work with. For example, we can characterize dosage by assessing cell count and viability for cell therapies and viral vector titer for gene therapies. We can perform an array of identity assays, including cell marker identification, gene expression, electrophoresis, Western blot, and flow cytometry-based assays. Similarly, we can perform diverse set of potency assays, including proliferation, cytotoxicity, and viral vector titer, as well as purity assays, including identification of residual host cell DNA and protein assays. Equipment highlights for analytical assays include the NC200, the ViaSol Blue cell counters, NovaFlex 2 metabolite analyzer, which can be very useful for upstream process development, the Beckman Coulter cytoplex flow cytometer, useful in cell marker identification, and infectious titer assays, and BioRad QX200 ddPCR system, which can be used for genomic titer assays. The Veriscan Lux plate reader for UV-Vis, fluorescence, the luminescence assays, and the ChemiDoc MP imager for imaging Western blots, SDS page, and nucleic gels assays. We also have a very strong leadership team with many years in the industry. Our core operations team has over 300 years of combined industry experience and continue to deliver as SMEs for our projects. Equally impressive is our advisory committee, which consists of big pharma, regulatory bodies like the FDA, equipment developers, as well as nonprofits. With that, I'll conclude. Thank you for your attention for our presentation. Please call us for your next project in cell and gene therapies.